All right, guys, today we're going to do a review of DNA. And I just want to warn you, DNA and evolution are a huge part of the EOC. So if there was one thing you need to understand, it would be this topic, okay? So DNA is the abbreviation for deoxyribonucleic acid, and that tells you a lot about the molecule right there. You have to know what kind of organic compound it is, which it is a nucleic acid. That's what the Na stands for. And as you know, DNA is every living thing's genetic code. Okay, so the building blocks of DNA are nucleotides. They're made of nucleic acids, like we just said. DNA shape is a double helix. Something that might be familiar to you would be like a spiral staircase or like a ladder if you could twist it. And the four nitrogen bases that are found in DNA are adenine, thymine, guanine, and cytosine. You do not have to know those. You can get through this test without it. You just have to know the first initial for it. Okay, and remember that A always pairs with T and C always pairs with G. So what we mean by that is how they discovered it is that if a cell was 22% A, it was automatically 22% T. That's how they discovered that they actually pair together like that, okay? And then the order of how your traits are determined is with these four right here, okay? DNA makes RNA, which makes your proteins, which determine your traits. So that's one of the things that you kind of have to know the order of, and you'd probably have to pick the right one. Okay, so if you guys remember, DNA has the instructions for everything going on in your body, and it stays safe and sound in the nucleus. So when it needs to tell the ribosome what to do, it has to make RNA, which is like a cousin of DNA. So this chart here kind of compares them. So DNA is double-stranded and RNA is only a single strand. So that's what allows the RNA to leave the nucleus when it is ready. The nitrogen bases, we've already said those for DNA, they're ATCG. RNA has a close cousin. Instead of it having a T, it has a U. So its base pairs are the same, except this time A pairs with U and C still pairs with G. Okay, so instead of doing where found, I want you to focus on the sugar. DNA is deoxyribose, that's why it starts with a D, and then RNA is ribose, that's why it starts with an R. All right, one of the skills you're going to have to be able to do is you're going to have to know the parts of a nucleotide. Okay, so anytime you see a sugar, that is a phosphate group. All right, anytime you see the pentagon, that is the sugar, which in this case, it's deoxyribose. If that were RNA, it would just be ribose. And then 2 here, that would be your nitrogen base, which could be ATCG, or if it were RNA, it could be a U. All right, and then there's a picture of DNA and RNA side by side, and you can kind of see the differences that we pointed out here in this chart. Okay, so guys, I'm sorry for the sloppy handwriting. I kind of did it in a hurry right before I started recording this. So you do have to know the structure of DNA. All right, so this is labeled for you, and I would like you to label it somewhere so you can kind of practice and remember it. So we did the nucleotide just a few seconds ago. That's your sugar, your phosphate, and a nitrogen base. So there are 4 different nucleotides depending on what kind of nitrogen base is attached to it. All right. The circle is always the phosphate. The pentagon is always the sugar. There will be no exceptions to that. I don't care what picture you look at on Google or any textbook. They will always be represented by those shapes. Okay. The letters are in between. Notice the base pairing that we talked about. That's something you will have to do on the exam. So if they give you TAAG, you should be able to say, oh, that pairs with ATTC. All right, that's one of the skills you have to be able to do. So this would be a base pair. Think about you have 2 shoes, right? It's called a pair. And then what holds the nitrogen bases together is a hydrogen bond. Hydrogen bonds are actually very weak, and you're probably thinking to yourself, why in the world would I want my DNA to be weak? Well, it's only because it has to be copied thousands of times a day. So it's relatively easy to unzip, so we can copy it so quickly. Okay? All right, familiarize yourself with that picture. All right, so then I left some practice things up here for you. Watson and Crick figured out the base pair rule, A with T, C with G. So if it asks you to find the complementary strand, you just put the opposite letters. And y'all, I kind of did it like this because I can't write as small as I can type. Okay, so you just put the base pairs next to it. Now, replication is when you copy DNA. So if you have a whole strand of DNA and that cell is getting ready to die or grow or whatever, the first thing you have to do is copy it. So that's what replication is. And it happens kind of just like what we did. The DNA molecule unzips, and then on the left side, A pairs with T, C pairs with G, etc. And on the right side, the same thing happens. So then eventually you will have two exact copies of DNA. Okay, the next part, this is where RNA comes in. Transcription is when you turn DNA into RNA. All right, remember there's no T, we have to do a U, but only when you want to say T. Okay, because one mistake my students do is if they see a T here, they automatically put a U. That's not how it works. Remember, A pairs with U. So whenever you want to say T, so for example, I would go C, C, G, O, T. Well, no, we're making RNA, so I need to write a U instead. All right? Then the next step, there's translation. That's actually when you turn RNA into a protein. And this happens at the ribosome, and you have to have the amino acid chart to do this. And I'm going to show you that on the next screen. Very important skill, so you need to make sure you know how to do that. Okay? So before we move on, just know that replication is copying DNA. Transcription and translation are the processes that turn DNA into a protein, and then those particular proteins make your traits. So for example, this one here that I created from these letters, and I'll show you on the next screen, okay, might be the characteristic for brown eyes, or for freckles, or for thorns on a rose bush, or whatever. Okay, that's what these amino acids end up turning into. All right? Okay, so guys, here is the chart like I promised you. This is the exact one that you'll see on the EOC. I took it off of a released one. You can actually see the word released in the background here. Okay, now, even if you don't know how to read this chart, I call it a chart for dummies, and I don't mean that disrespectfully, because what they do is they actually put the codons here. All right, the ones I learned on and had to memorize didn't have those. So anyway, what you do is, let's say I'm trying to figure out CCA, okay? So the first letter tells you what row it's in, all right? So for example, let me see if I can do this with the highlighter for you. Okay, the first C tells me that my answer is going to be somewhere in this row right here, okay? Then the second C tells me what column it's in. So I'm going to use a different color. Here's this column. All right. So that tells me my answer is somewhere where the pink and the yellow overlap. So the good news is, for this particular amino acid, I don't even need the A, because every amino acid in this particular box is proline. So CCA would be proline. Okay? All right, so let's reset that. Let's try one more. Let's do G, let me make caps, GCA, okay? So let's do that again. G tells me that it is in the bottom row, okay? The C tells me it is in this column, all right? And again, I don't need that third amino acid because all of the four in this particular box are alanine. All right, okay, I'm going to do one more so I can ensure that we have something different here. Let me do UGG. Okay, all right, so I happen to know this one because I think of the UGG boots that people have, and I've memorized this one. All right, so the U tells me it is in this top row here. All right, then I need to find the G. That tells me it's somewhere in this column. So now I have 3 options here. It can either be cysteine, stop, or tryptophan. Well, the last letter is G. Here's a G. So I do indeed need the tryptophan amino acid. Okay, so guys, that's how you use that chart. It's actually very easy, but you can see if I was doing UGG, I could find just UGG right there and go with tryptophan. All right, so don't feel like you're lost if you don't know how to do it the way I just showed you. Just look for the correct amino acid. Okay, so guys, this picture kind of shows all of that in a nutshell. So here's DNA. We know that because it's labeled, but you also know that because it's double-stranded. Okay, transcription is when we turn DNA into RNA. Notice it's a single strand, okay, and it has different colors in here to represent the U instead of the T. It heads over to the ribosome. The ribosome reads the RNA in sections of 3, which is called the codon, okay? These guys here that look like little coat hangers, and I actually like these because they kind of look like the letter T, okay? These are tRNA molecules. And what they do is they carry the correct amino acid and put it in the right spot. It's like a very good waitress. If you order something, a good waitress can say, here's your shrimp burger, or here's your steak and fries, or whatever. Okay? And then they go off and pick up another one. So then this is your growing amino acid chain here, which is a protein. And then once it's done, it gets cut off and it creates the trait that you were desiring there. Okay, all right, I just want to mention something super quick. Mutations will be on there, not necessarily the specific kinds, but what can happen. Okay, if there is a single base pair changed on the DNA, then it is called what is it called? That's just like, it could be a mutation because you could have one base turn into another one. Okay, that's called a base pair mutation, and that could be something as simple as a new amino acid being made, or it could be no change at all, which would be wonderful. And these other things here are mutations that happen on your chromosomes, and this one here, of course, is the worst one. And then I do want to take a minute just to talk about DNA technology, because I'm pretty sure that would be on there. We spent a couple lessons working on gel electrophoresis, which is when you run electricity through your DNA and it makes these bands, okay? And just remember, if you're trying to find a criminal, the lines should match 1 for 1 for 1. So for example, the blood stain matches John, so he would probably be the suspect in this case. Okay, if you're matching for parents, half of the line should match the mom and half should match the dad. You can also use this to figure out common ancestors. So the more lines they have in common, the more closely they're related. All right, then finally, this is some DNA technology, some DNA fingerprinting here. Not fingerprinting, sorry, some genetic technology. So what we can do with this is we can actually get bacteria to make things they normally don't. So an example of this is insulin. So instead of taking the insulin from pigs after they've been slaughtered and using their pancreas to get like a teeny tiny amount of medicine, we can get bacteria to make insulin for us. They've done this for lots of different hormones, but insulin is a very good example for that. So if you see this, I want you to pick genetic technology, okay, because you're getting, or genetic engineering, because you're making the bacteria do something that it never did before. This is just an application of fingerprinting, which means we can find criminals or baby daddies or different things like that, okay? All right, so I'd like y'all to do the practice that I've assigned, and make sure if you have any questions, you contact me. Like I said, DNA is very heavy on the exam, so make sure you're comfortable with it. Thank you.